All right. So in this short lecture, what I want to talk to you about is our neuron model here. And so what we're going to do is look at some of the organelles and see how this relates to the central dogma of biology. Okay, so the first question that I have for you is, what type of cell is this? Is this a specialized cell, or is this a generalized eukaryotic cell? It's a specialized cell. And what is the particular type of cell that I have here? Neuron. It's a neuron model. Okay? And so what I want you to understand is that we use the generalized eukaryotic cell, and we can see that the processes are fairly similar here within this specialized cell known as a neuron. Okay? So the first thing that I want to talk about is the central dogma of biology. So the central dogma of biology states that we go from DNA to what? Messenger RNA. And for messenger RNA, we make protein. And so the first process that can happen if we replicate DNA, so number one, this is DNA replication. Number two, going from DNA to messenger RNA, is this transcription or translation? Transcription. So 2 is transcription. We're making transcripts. Okay, so number 3, from messenger RNA to protein, what is this called? Translation. All right. So another thing that I want to discuss here is, where do these steps take place? Where does DNA replication happen inside the cell? In the nucleus. Excellent. Where does transcription occur? In the nucleus. And where does translation happen? In the cytoplasm. And where within the cytoplasm, well, besides where, the first thing I'll ask is, translation occurs on what type of organelle? Ribosomes. And where are ribosomes found? Exactly. So we have the rough ER, and then also we have our unbound ribosomes that are just found within the cytosol. So these are the two locations where the ribosomes are found. So is everyone clear on this? This makes sense? Yes. Okay, excellent. So I want to point out also, with transcription, what do we need in order to have a mature mRNA strand? So to have a mature mRNA, what are some of the components here? We need 3 things. We need the 5' cap. We need what? The poly A tail. And then what do we have to splice out? Introns or exons? Yeah, so we splice out introns and we express the exons. All right, so these are the 3 conditions that must be met to have our mature mRNA. And what are some of the components of the ribosome? What are the two subunits? So if I draw a ribosome here, we have so this will be 1, we'll make this A and this B. So for A, we have a large subunit, and for B, we have a small subunit. So what will happen, that messenger RNA will get here into this ribosome, and what do we form? So whenever translation happens, we said that we make proteins, right? And what are proteins composed of? So proteins, when we say a protein, what we're referring to is a polymer. What if I say, what is the monomers that make up proteins? Amino acids. And some of the functional groups for amino acids, so this here is my central carbon, and I have this functional group here, and then I have another functional group here. So this part of the amino acid, this is the amine group. And this part over here, this is my acid. More specifically, a carboxylic acid. And this R group here that's found on the bottom, so I can write, so I'll write the R group. So for the R group, we have various types of amino acids, okay? And there's a couple different groups of them. So we have nonpolar amino acids, we have charged amino acids, so they could be positive or negative. We also have what's known as aromatic amino acids. So what I want to point out to you is that this R group here is going to determine how the amino acid behaves, how it folds, how it interacts with the environment. Okay. So the next thing, we've said that this is an amino acid, and these amino acids are linked together to each other, right, to form this long chain. So the next thing I want to point out to you, let's see here, I'm going to go on this side. I think I have. So this is a dipeptide. This means that I have two amino acid residues here. Okay, so the first thing I'm going to draw is the carbon skeleton, and then this is the R group here that's on the bottom. So now let's draw, this is called the N-terminal of it. So the N-terminal will have the amine, the C-terminal will have the carboxylic acid on the group. Everyone good so far? Yes. Okay, so this part right here, I'm going to finish drawing this out, but this is what's known as a peptide bond. So the peptide bond is what links amino acid residues to form our peptide or protein. And so at this N-terminal, we have this, and then we have this. See that? Because this can be cleaved, and we can break this apart through a process known as hydrolysis. But I just want to point out to you how the amino acids are linked together. So this is they're linked together by what's known as a peptide bond. Okay, so now let's go back to this stuff. So we went over some of the basics. So now let's apply this basic principle to our neuron here. Okay. So I'm just going to leave this down for right now. It's hitting the mic. Okay. So this we've already established. We've learned today in lab, right, that this is the nucleus. This part here in the middle, this is the nucleolus. What's assembled here in the nucleolus are the ribosomes. So this is where the manufacturing of them happens. Okay, so for transcription to happen, we find this particular sequence on DNA that codes for the messenger RNA. And we make our transcript. So we'll make our transcript, and how does that transcript get out of the nucleus? We have these little pores here. These little pores are nuclear pores. So that mRNA transcript will get out. It'll get out of the nucleus. And where is it typically going to go? These little studs on this, what is this? Is this rough ER or smooth ER? This is the rough ER. So this is where these ribosomes are located. And because this is a specialized cell, we can synthesize what's known as neuropeptides. So it's a specific type of peptide. Okay? So we'll synthesize our neuropeptide here, and then it'll get packaged in this vesicle. So this vesicle will bud off, and then it gets to this green structure. What is this green structure known as? The Golgi. So this is the Golgi apparatus. And what is the function of the Golgi apparatus? What is the basic thing that they tell you? What do they always say? It's it's where the packaging occurs. We call it like the UPS. So we're packaging that neuropeptide into a vesicle. Okay? And we're getting it in this vesicle because this neuropeptide, so a neuropeptide is a type of neurotransmitter. Neurotransmitters, that's how neurons communicate with each other. So we need to get this neuropeptide all the way down this axon. This is called the axon of the neuron. Right? Because this is how, like, one neuron here will synapse with the other one. Okay, but how do we get this vesicle that contains these neuropeptides, how do we get it all the way down? Well, once this vesicle will leave, it travels along here within the axon. Within this axon, this is where the microtubules are. So you could think of them, they're kind of like train tracks, I guess you could say. So that vesicle is going to package that neuropeptide within that vesicle, and then it's going to travel along the axon. So it'll travel along the axon, and then it'll get to the axon terminal, and it'll kind of just sit there in that vesicle. So how neurotransmitters communicate, action potentials will be submitted back and forth, and if the axon terminal receives the signal, it'll release the neurotransmitter or the neuropeptide that we just discussed, and to communicate onto the next neuron. Good? Yeah? All right. So are there any questions regarding the neuron in relation to the central dogma of biology? No? All right, so that's it for this lecture.